Thanks to the organizers, Wolfram and Jay. Thanks to the chairs for introducing me. And thanks for letting me give a talk about our recent results in repairing Duchenne muscular dystrophy and some other things which you will see in a minute. So, if that moves. Duchenne muscular dystrophy is a devastating disease affecting 1 in 3,500 boys in Western countries. Okay, now it goes. Which leads to a loss of ambulation early on, then a respiratory weakness later, cardiomyopathy occurring in the second or third decade of life, and fatal arrhythmias being not uncommon, in the end leading to premature death. Reason for that is the absence of dystrophin, the second largest protein, napstitin, in the human body, which, due to its sheer size, gives ample reason for mutation, which occur throughout the whole length of the gene sequence and are modeled in a variety of mouse and dog models, and the number is increasing. Why would we need gene editing of Duchenne? There is antisense nucleotide treatment which is approved, at least conditionally, however needing a frequent repetition of the treatment and the protein expression in the biopsies appearing rather weak, as is the functional effect. AAV supplementation therapy with mini or microdystrophins is in clinical trials right now. However, again, it would be a temporary therapy, and reducing would be questionable due to the rise of neutralizing antibodies. And finally, permanent correction of the muscle and, if possible, satellite nuclei would be desirable to cure the diseased muscle cells once and for all. We tried that in a large animal model in vivo, and we used the properties of Cas, providing double strand breaks in mammalian cells, which then are repaired by the cell's own repair machinery, often by insertions and deletions, which could also be provided by zinc finger or TALEN proteins, which, however, due to their nature of protein-DNA interaction, are much more restricted than the RNA-DNA interaction provided by the guiding principles leading to Cas-mediated breaks. Now, this has been tried in oocytes in the lab of Eric Olsen early on, where the MDX spontaneous mutation in the mouse world was treated with Cas and led to a mosaicism or a perfect correction on this level, in the end just creating a transgenic mouse. But this was only the prelude to a more therapeutic experiment where adult mice were treated systemically with the two-vector approach, coding for Cas9 and a gRNA, single guide approach, which not only provides ample transduction of the myocytes, but also re-expression of the red colored dystrophin in muscle and heart cells, increasing over time. And not only that, but also in a large animal, the Cavalier King Charles Cocker Spaniel, lacking exon 50, there was the attempt to excise exon 51, allowing for the expression of a shortened but functional dystrophin protein, which succeeded not only with regional injection of the Cas9 and guide RNA combination, but also with high-dose intravenous injection, leading to a wide variety of muscle re-expression of dystrophin, which is absent without any transduction. Now, we had the privilege and pleasure to work with, okay, I don't know why that is, but there is something wrong with the pointer. We had the privilege and pleasure to work with Eckhard Wolf and Nick Klumyuk in Munich to nurse a pig which was lacking exon 52, which was replaced by a reselection cassette. This was done in fibroblasts of the pig, then a nucleus was taken to the oocyte, and then a new pig was generated by the Dolly method, as seen here, enucleation of the oocyte, renucleation of the chosen fibroblast nucleus, electrical activation, and then release of the embryonic program. This worked, as you see, in this picture, where as opposed to a wild-type pig, which is happily overcoming small and larger hurdles, the DMD pig has some trouble and clear impaired motor behavior. Now, we wanted to excise the adjacent exon 51, leading to a shortened dystrophin protein lacking exon 51 and 2, and this happened on the genomic level, as seen here in the intramuscular injected pigs with a high frequency, but also in high dose intravenous treatment in a variety of muscles. This is confirmed on the protein level. Here again, the intramuscular injection, and here the high-dose intravenous injection. And also on the histological level, here you see the wild-type pig, Duchenne pig without any dystrophin unless it's injected regionally or high-dose intravenously. Principal component analysis of the proteome indicated that the treated muscle reside more to the normal muscle phenotype proteomic expression as the untreated muscle, and video footage of 24-hour observation period with color coding for lying or sitting posture in blue and action in orange indicated that, as opposed to the wild type with the plentiful activity, there is a more resting state for the untreated Duchenne animal, which is corrected, at least in part, by high-dose intravenous treatment. Quantification is here and is juxtaposed by the levels of creatine kinase in the blood, which is an enzyme for muscle decay. Now, this video footage also allowed for observations, which I want to share here. I want you to focus on this guy particularly, which playing happily with its peers until it all of a sudden drops and never rises again. And this is the feature of sudden cardiac death, which none animal of the cohort we observed survived after day 105, a day which at least was surpassed up to 1/3 when we treated the animals with high-dose Cas9G RNA AAVs. We subjected some of the animals to high-resolution electrophysiological mapping, which was introduced to you before in this meeting, where violet is a normal action potential amplitude, and red is none, being basically fibrotic scar, and green, yellow, and blue being in between. Here you see in the wild-type plentiful violet, there is a normal action potential throughout the left ventricular muscle, which is also happening in the Becker phenotype, a pig where we, in the oocyte, also excise the exon 51 in addition to exon 52. There's an almost normal heart. This is not the case, however, if you look at the DMD pig, where there's a large area of either fibrosis or low amplitude, which is a risk area for origins of malignant arrhythmias. And that can be at least reduced by high-dose intravenous treatment with our Cas vectors systemically. Now, the same could be replicated in a surrogate experiment with patient-derived cardiomyocytes, the patient lacking also exon 52, where upon the two-vector approach correction in the calcium transients of the individual cardiomyocytes, here's the wild type, the Duchenne, the Becker phenotype, and here the treated cardiomyocytes, you see a normalization of the length and the width of the calcium amplitude, and the triggered events as a surrogate parameter for arrhythmias also clearly present in the patient's cardiomyocytes were disappearing upon correction either on the IPS or on the cardiomyocyte level, indicating that gene editing by Cas and gRNAs induces expression of a shortened but stable dystrophin protein, which can avoid sudden cardiac death, the leading cause of limited lifespan in pigs. Now, one more thing. In these animals, we took the apex of the heart, cut it and put it on a biomimetic chamber and superseded it with human ventricular progenitor cells. And upon this measure, the decay of muscle force development in the biomimetic chamber from day 14 on was largely avoided. Now, what are these cells? These are from human embryonic pluripotent stem cells. They are generated by standard protocols, and from day 6 on they are called human ventricular progenitors because they don't express mature genes already, which however happens standardly or systematically over time, and from day 14 on they are called ventricular myocytes. These were, in a set of experiments, superseded on these heart slices in biomimetic chambers. And 3 days later, which is called day 0 here, a radiofrequency ablation injury, like electrophysiologists do to cut conductance muscles in the heart, were applied. And you see here that the RFA injury at day 0 is dark, but on day 4 already, there is a clear movement of the NKX GFP positive cardiomyocytes is occurring, which is absent in experiments with mature cardiomyocytes, and which is depending on CXCR4, since the blocker of this molecule, AMD 3100, was completely avoiding the migration to the RFA lesion. In opposite, the CXCL12 ligand abundance in the Boyden chamber led to an increase of migration towards the ligand, such that the SDF1 or CXCL12, CXCR4 axis is established as an essential element for the migratory behavior of human ventricular progenitors in ex vivo experiments. On the other hand, fibroblasts populate the RFA lesion first. And these fibroblasts were put as monolayer on slides and again superseded with the HVPs and again injured by the radiofrequency ablation. And here you can see that again these human ventricular progenitor cells march towards the lesion and on the course repel the fibroblasts from the lesion which they want to populate. And this behavior is dependent on a robo-slit interaction, since an antibody against robo avoids that, and application of the recombinant SLIT1 increases that behavior. That may explain the pictures you see here. The injury at day 1 is empty, at day 7 is filled with fibroblasts, and at day 21 is filled with myocytes, which derive from the ventricular population, which on the way repels the fibroblasts from the injury and puts them to the border zone. Now, would that happen in vivo? For that, we designed a pig experiment where we epicardially ablated myocardium, radiofrequency ablation, and put the cells 1 centimeter beneath with a syringe, and then observed the animals up to day 14. And you may appreciate that from day 3 on, the cells moved from the syringe channel towards the site of injury and are again depending on the CXCR4, CXCL12 axis, since the blockade in vivo in the animal would avoid the reaction. And scar formation is again diminished by this behavior. Last data slide shows that in a more clinical set of experiments where 3 weeks after LAD occlusion, permanent occlusion, Cells were applied at this dose, 1 × 10⁹ HBPs, in the vicinity of the myocardial infarction scar. And then after 3 months, there was an analysis of tissue and function. And you may appreciate that there are clusters of cells in the infarcted zone, that the function in global longitudinal strain is maintained better than in the control experiments, and the infarct scar is diminished significantly by this intervention. I may summarize that the cascade of developmental programs of these human ventricular progenitor cells reaching from sensing phases towards migration on the CXCL4, CXCL12 axis, and repulsion of fibroblasts with the virtue of SLIT2-ROBO1 interaction leads to filling of the injured myocardium with replenished human ventricular progenitor cells, contributing to scar reduction and functional improvement. This has been shown ex vivo in heart slices and in 2 independent models of in vivo experiments. I don't want to stop without thanking the many people contributing here, from my lab, the Moretti-Laubwitz lab, the Wurst lab, the Wolf lab providing the animals, collaborators extramurally and intramurally, funding agency. Thanks for your attention.